In this lecture, we will talk about niacin, also called B3. There are a few different subforms of niacin which we refer to as vitamers. Nicotinic acid is one, which you can see on the upper left of the screen. So this is a pyridine 3-carboxylic acid. This is often, when we say niacin, is what most people are referring to, but there are other forms that others kind of collectively call niacin. So this form has a carboxylic acid, so a carbon, 2 oxygens, and a hydrogen attached to the nitrogen ring structure. There's also nicotinamide, which in the place of the alcohol group has an amide group, NH2. And there are other nutrients that we can derive niacin from, things like tryptophan and nicotinamide riboside. Now, niacin is a much more simplistic molecule, which we saw on this slide. But in order for this to actually have activity biologically in our bodies, we need to convert it into the active coenzyme form. And so this would be NADH or NADPH. And so you can see at the very top, highlighted in the yellow square, that's nicotinamide, and that's where the reactive site is contained. So this is where niacin can carry out its metabolic activities. But that has to be connected to a ribose and then an adenosine. And this basically then primes or energizes this molecule to be able to perform biological functions for us. Now this is too big for us to absorb, so we would have to break this up if we were to consume it into the nicotinamide or nicotinic acid. And then those would be absorbed and we would rebuild this molecule. You can also see down at the bottom I have a hydrogen marked in red. So this is the location where we can slightly alter this structure to allow it to operate in different metabolic pathways. So at this location the hydrogen could be removed and we could add a phosphate, for example. So then that's how we would create NADP or NADPH. And then we could remove that phosphate and put a hydrogen back on, and then we would be working within the constructs of NAD, NADH. And we'll look at some pathways that each of those are involved in later on. So both of these vitamers or coenzyme forms become really important for different pathways. You can see that there's a subtle difference between the two. So here's that hydroxyl group we saw in the previous slide, and we can switch out the hydrogen for a phosphate, and that's the only difference between these two molecules of niacin. The primary way that niacin will act as a coenzyme in the body is by aiding redox reactions. So this is all about moving electrons, which we often illustrate by moving hydrogens. So NAD plus is not fully endowed with as many hydrogens as it can carry. So we see on this top carbon of the ring structure, there's one hydrogen, and then that can have a hydrogen and electron added, and we end up with 2 hydrogens, and that's what we call NADH. And this can easily toggle back and forth between the two reactions. So if we're following the path from left to right, this would be reduction of NAD, so it's gaining an electron. We see that by adding that hydrogen. And it's obviously having to interact with another substrate in order to gain this electron. And that substrate that it interacts with is being oxidized. So it's giving up an electron that's then being added to NAD+. In the reverse, if we go right to left, the NADH will be oxidized. It will lose an electron, and the substrate interacting in this reaction would be reduced because it gains that electron. So in our tissues, both NAD and NADPH often tend to move in the left to right or right to left direction for the reaction on this slide. So NAD is often involved in oxidation reactions where a substrate is being oxidized and NAD is gaining that lost electron, whereas NADPH is often present in the NADPH form, so it is going to lead to reduction reactions where a substrate is reduced and NADPH gives away its electron. That's not always true, but in many reactions within the Krebs cycle or those reactions connected to the Krebs cycle, this is sort of, you can bin these two molecules into these categories. Now, where do we get niacin? Well, it is present in a variety of food sources, and so I went to the USDA database of foods, and I just pulled up the top 10 dietary sources in order of niacin endowment. And so you can see that beef liver is the most rich, providing 93% of our daily value at just 3 ounces of product. There's quite a few meat products here, or protein-based products, so chicken, turkey, salmon, tuna, pork, beef. And then we see a few other things sprinkled in, like marinara spaghetti sauce, so coming from fruits, and then also rice and breakfast cereals. And their niacin is pretty well provided across the different food groups, but you can kind of gain an appreciation for those if you really want to get more bang for your buck to get lots of niacin in. That can happen with animal products, and grains, I would say, are the two major categories here. So when we're talking about animal-based foods, the niacin is usually already formed into the cofactors that had the ribose and the adenosine connected to it, so as either NAD or NADP. The liver's a major site in our body that can actually convert tryptophan into NAD. This reaction is also dependent on a number of other B vitamins, specifically riboflavin and vitamin B6, as well as iron. So, in theory, a niacin deficiency could actually be caused by a deficiency in riboflavin, B6, or iron, and sort of mask the niacin. Because if you can't convert tryptophan to niacin, then you may not be able to achieve the amounts that you need. And so evolutionarily, our body has found a way to convert tryptophan to niacin to ensure that we have adequate endowment of this nutrient. And so for every 1 milligram of niacin that we need to produce, it takes 60 milligrams of tryptophan from our diet. One interesting fact about that is if you only look at the food composition tables, so on the top 10 list of foods that I showed you on a previous slide, that's only showing preformed niacin. It's not accounting for the additional niacin that we could derive from tryptophan, particularly in all of the protein source foods. So as a dietitian, you may need to account for tryptophan conversion. If you suspect someone is possibly deficient in niacin, you might also want to just see, oh, well, they eat a really high protein diet. They should, in theory, be making niacin from that. And if they're not, then that might indicate there's some sort of defect in a gene that's converting, or a deficiency in either riboflavin, vitamin B6, or iron. Now, plant foods, on the other hand, are often bound a little differently. So we don't often find them as NAD or NADP. They will be covalently bound to complex peptides or carbohydrates, and so we call these niacinogens and niacetins. And when we eat plant-based foods that have niacin in them, it's actually much less bioavailable because the niacin is bound to these carbohydrates and peptides. Now, there is a way that we can process the food in a way that will free more of the niacin. So we can free about 10% of the niacin that's bound to niacin or niacinogens by treating the food product with either hydrochloric acid or lime, which is essentially like bicarbonate. And this is a really interesting sort of practice that's been developed over time. So corn masa, for example, is often treated with lime prior to making tortillas and other corn-based products, because this frees the niacin, and it frees enough that it can maintain human recommended needs. One interesting story about that too is that when Christopher Columbus came over to the U.S. And discovered the indigenous populations here and learned of how they processed corn, and I guess he didn't really pay that much attention to actually how they processed it, so he brought a bunch of corn back with him to Spain, and niacin deficiencies became prevalent in certain areas because they weren't processing the corn correctly in order to free that niacin from the plant. Oh, and then supplements, those are usually going to be nicotinamide. This is the general form, but that's not guaranteed. It's just historically that's been probably the most common. So let's take a look at how tryptophan is converted into niacin. And just as an aside, I won't expect you to know each step in this pathway, but I have 2 sections highlighted in blue that are important because they involve some of those cofactors that are required for converting this amino acid into a vitamin. So the liver has a variety of enzymes that can take tryptophan, and the very first step is iron dependent, and subsequent steps use both B6 and riboflavin in a repetitive process to trim down the molecule into what will become the ring structure with one singular carboxylic acid on the side. So this particular pathway accounts for about half of the niacin that a human needs on a daily basis. So we end up utilizing a lot of the tryptophan that we consume from protein-based foods to maintain niacin status. And so if someone wasn't getting a lot of preformed niacin and they were dependent on this tryptophan conversion, then they really also have to have iron, riboflavin, and B6 present. They also need to make sure that they are eating tryptophan as a replacer. And so this is what I meant when I said that a deficiency in iron, riboflavin, or B6 could mimic or mask a niacin deficiency. And so you sometimes have to check multiple nutrients for their status in order to confirm which nutrient is the culprit and which foods need to be ensured that they're added to the diet to compensate for the loss or the deficit. For digestion and absorption, niacin is absorbed through the majority of the small intestine. And so here you'll notice that I have sort of highlighted in pink the 3 sections of the small intestine. So these are the duodenum, the jejunum, and the ileum. And then I also show the colon, which we don't see any pink. So that means that we really don't absorb niacin in the large intestine and colon. And so to give you a sense of what the digestion process is, you can generally see that depending on what you've consumed, there may be some different steps involved. In the duodenum, we have NAD or NADP. So this has that ribose sugar and the adenosine phosphate that have to be removed either by a pyrophosphatase or a glycohydrolase enzyme, and that will free the nicotinamide. And I just imaged this as a single ring structure without the arm. And then that will get absorbed through transporters that are also sodium dependent, hence the presence of sodium. Now, some foods just have the niacin, so they would go straight down to the location of absorption and co-transport with sodium. Looking at this pathway in a slightly different way with a little bit more detail, here we can see that depending on if we consumed foods or supplements, nicotinamide, nicotinic acid, NAD and NADP are all coming in, and the NADP will have to have the phosphate first cut off by a pyrophosphatase. That leaves us with NAD, and that's where the glycohydrolase comes in, so cleaving off those ribose and adenosine components. And that leaves us with either nicotinamide or nicotinic acid. More commonly, it will be nicotinamide, and that gets absorbed through the intestinal epithelial mucosa through a transporter and then sent out into the portal blood for transport to different tissues. Small amounts of nicotinic acid won't be absorbed, and they will travel all the way through the intestine and be excreted in the fecal matter. Overall, we're really good at absorbing niacin, so 100% is absorbed with up to a 3 to 4 gram dose. So you can consume quite a bit before you would lose efficiency in your absorption rate. At low concentrations, we absorb through that sodium-dependent process. So this involves a protein carrier to help create a portal for the niacin to move through, and sodium moves with it. This is not energy dependent, so we would call this facilitated diffusion. And then at high concentrations, the protein carrier still helps to move niacin, but our body operates on concentration gradients and trying to equilibrate the amount of a substrate on both sides of a membrane. So when you consume a ton of niacin, it will actually passively diffuse. So it will just move through the membrane without the help of a protein to try to balance the concentration on both sides of the membrane. And so at high concentration intake, then our bodies use more passive diffusion. So transport and storage are, once we've digested and absorbed this nutrient into the intestinal cell and it gets out into the blood, it's going to travel in the plasma to tissues in need. So niacin is primarily found as nicotinamide. That can move across the cell membrane on the basolateral membrane side by simple diffusion. So on the luminal side that lines the intestinal lumen, that can have a carrier, but then we move kind of to more simple diffusion once we're transferring into the blood on the other side of the cell. The portal blood often goes and delivers these nutrients absorbed to the liver, and it's there that we can synthesize either NAD or NADP, and any excess amounts can be converted to nicotinamide if we need to have them that way, which would then be available to transport to other tissues. So this means the liver takes what it needs first, and any excess will be converted to nicotinamide for transport to, say, the brain, the heart, muscle, wherever it needs to go. NAD and NADP, once you've added that ribose and the adenosine and phosphate, they get trapped inside a cell. So the coenzyme is basically prepared by the cell, which is nice for the cell because it's like, okay, well, I made this coenzyme and now you're trapped here because you're too big to get back out into the blood, and so I'm going to hold on to you so that I can use you when I need you. And of course, once the cell decides it doesn't need it, it can easily cleave off the extra parts, and then the niacin can go back out into the blood. So that here's a pictorial illustration of what we've been talking about on the last few slides. So in the blood, we have transport of niacin. In the portal system, it is released into the liver. The liver can take whatever it needs and store it, lock it inside the liver cells by adding ribose and adenosine and or phosphate. And then if it has excess and can share with other organs, it will cleave off those extra parts. And both, so if it produces niacin, then that could travel, say, to the muscle, which will then lock it inside the muscle cells through the same pathway of adding ribose and adenosine. The liver is also the central site of excretion. So if we go back to the liver, we can see an excretionary product, which is not fully cleaved, but still contains a methyl group and another ring structure. So we call this N-methyl nicotinamide, NMN, and so this basically primes the molecule for excretion in the urine. And then that molecule will travel to the kidneys for excretion. Now, niacin is active in nearly every tissue in the body, anything that utilizes energy in some way, so that's basically every cell. So more than 200 enzymes utilize niacin as a cofactor. Mostly these are dehydrogenases, things that are involved in redox reactions. And those redox reactions allow for the synthesis and degradation of all of our macronutrients, so carbohydrates, fats, and proteins. So one of the major things we do is we oxidize fuel molecules, so glucose and fatty acids, to generate energy. And whenever we are doing that in our cells, niacin is there to help. Because the energy and fuel is so important to maintaining all of our daily biological and metabolic functions, niacin will tightly bind to its enzyme partner, which we call an apoenzyme, and it can easily then transport hydrogen atoms from one part of a cell to another. So basically, in fuel utilization, we're talking about taking a hydrogen released from a Krebs cycle reaction and moving it over to the electron transport chain in a different location within the mitochondria. And while redox reactions are sort of the primary function of niacin, there are non-redox functions. So niacin can also be a donor of adenosine diphosphate ribose. And this becomes important for components of building new DNA strands, activating certain proteins, and helping with the processing of certain chain-length fatty acids. So in essence, we can break niacin's functions and mechanisms down into two main categories. So we have our NAD pathways and our NADP pathways. The NAD plus NADH pathways are basically exclusively involved in electron transport. So NAD plus acts as an electron acceptor. It takes 2 electrons and a hydrogen or a hydride ion. These activities are tied to dehydration reactions. So dehydrating means removing hydrogen from something, and then the NAD collects that hydrogen that's been removed. And one of the primary places that NAD shares those captured hydrogens is the electron transport chain, which generates ATP by transferring electrons from metabolic intermediates like glucose and fat. NADPH is involved in what I categorize as biosynthetic pathways. So this is where we're building new molecules. NADPH will act as an electron donor. So NADP often already has the H on it, and so it's giving the hydrogen to something else. This is really helpful for building fatty acids. It's really helpful for building cholesterol and steroid hormones. It helps us to regenerate important antioxidants in our body like glutathione, vitamin C, and thioredoxin. And it's also involved in the conversion between different forms of folate. So let's take a minute just to review oxidation reduction reactions. These are things that you cover in biochemistry, and they're often abbreviated redox reactions. The red part of the word refers to reduction, and the ox part of the word refers to oxidation. This is basically the gain and loss of electrons, and you may be familiar with the mnemonic device oil rig. So oxidation is loss, reduction is gain. And there are quite a few enzyme class categories within the oxidoreductases which carry out redox reactions. So these can be oxygenases, reductases, peroxidases, oxidases, hydroxylases, and dehydrogenases. And dehydrogenases are one of the major ones that we're going to cover in this class. So let's look at NAD plus being active in a dehydrogenation reaction. So here we have an oxidation reaction. So a substrate is going to lose 2 electrons and a hydrogen. NAD is going to accept one of those electrons to become NADH, and then we'll have that free-floating hydrogen that was also released along with that in solution. And then our substrate becomes substrate 2 because it lost electrons and hydrogen. And of course, the enzyme carrying this out is a dehydrogenase, meaning it removed a hydrogen from the substrate and gave it to NAD, its cofactor. Oxidation reactions like this occur all throughout human metabolism, including glycolysis, the Krebs cycle, and ultimately passing these electrons to the electron transport chain. So some specific examples I have listed at the top of the slide. So these would be oxidative decarboxylation of pyruvate to acetyl-CoA. So that we can see that at the bottom of the slide. Pyruvate is the final product of glycolysis that passes into the mitochondria to become acetyl-CoA for integration into that pathway. We can also oxidize acetyl-CoA to the next substrate, which is citrate. And then we have beta fatty acid oxidation, so that's helping to break down fatty acids to feed into the Krebs cycle. And we also use redox reactions when we metabolize ethanol. So again, NAD in a dehydrogenation reaction is all about transferring electrons from a substrate to an electron acceptor. That electron acceptor is NAD. And the enzyme helps to facilitate that activity by binding to the NAD and working together. So here we have our substrate. We can see the hydrogens that are removed from it. One of those hydrogens is added to NADH, so our substrate is oxidized while NAD was reduced to NADH because it gained that electron while the substrate lost electrons. NAD is involved in 2 dehydrogenase reactions in the glycolysis pathway. So this is the glyceraldehyde 3-phosphate dehydrogenase step. This is an aerobic process requiring oxygen in order to carry out the reaction. And then also tied to glycolysis, we have lactate dehydrogenase, which is an anaerobic pathway not requiring oxygen, which NAD also assists with. For beta fatty acid oxidation, NAD is reduced to NADH, just like in the glycolysis and Krebs cycle reactions. And then the NADH can move over to the electron transport chain to donate those hydrogens and electrons. Activated fatty acids, which we're seeing here, are processed to create acetyl-CoA, which can then be shuttled into the TCA cycle for ATP generation. So this is essentially how we're able to use fat in order to make energy for ourselves in a shortage of glucose. And then finally, looking at the TCA cycle, I'll just highlight a few of the reactions that utilize NAD. So we can see there are several locations. We have the pyruvate to acetyl-CoA conversion, and that's what allows us to then fully move into the TCA cycle. Isocitrate to alpha-ketoglutarate and then succinate, both require NAD, and then again towards the end of the cycle, malate going to oxaloacetate. And one of the locations where fatty acids would then be entering is at acetyl-CoA, and that again requires NADH as well. And then we have all of those electrons from each of those reactions that are then carried to the intermembrane space of the mitochondria, where the electron transport chain proteins reside. And so we can zoom in on that location. So here in the mitochondria, we see that we have the outer and the inner bendy membrane. And so if we take a picture of just that one little spot where we have the outer membrane and the inner membrane, we can see that the electron transport chain proteins are embedded in the inner membrane. And essentially what these proteins' job is to take electrons from a low energy place, so the mitochondrial matrix on the inside of both membranes, and pump those hydrogens and electrons across to the high energy space between the two membranes. And so electrons are used to fuel energy across these proteins to keep pumping the hydrogens across. And ultimately, those hydrogens collect in such a high number in this space that that's able to fuel ATP synthase to take ADP and add a third phosphate to produce ATP. Now, I mentioned that ethanol is also metabolized through redox reactions, and I wanted to show you some of the steps involved in that utilize NAD. So in order for us to metabolize alcohol, there are a few steps. So the first step, if you have a low intake of alcohol, you use alcohol dehydrogenase in liver hepatocytes. This happens in the cytosol of the hepatocytes. So if we were to look at where that is happening in the graphic below, it's this middle frame. So this is our ethanol. Then we have ADH with NAD as a cofactor, producing acetaldehyde in the cytosol. Acetaldehyde can be shuttled into the mitochondria then for the second step, where ALDH2 or alcohol dehydrogenase 2 works to further convert it into acetate with the help of NAD. So that's at low intake. If you have a high intake, we require other mechanisms to more rapidly get rid of ethanol in the body. So liver hepatocytes and the endoplasmic reticulum within those cells have a different enzyme called cytochrome P450 2E1. And so that's CYP2E1 down here. So this is at the microsome, so basically the endoplasmic reticulum. And NADPH will help that enzyme to break down ethanol into acetaldehyde faster to keep shuttling it into the mitochondria, after which. We can also use catalase, which is utilizes hydrogen peroxide to take ethanol and turn it into acetaldehyde within peroxisomes. And these outer two darker gray boxes only occur if we have really high intakes. So they're not really the preferred method. they're more labor intensive, and they do produce sometimes adverse oxidants like hydrogen peroxide and other free radicals. One thing that can occur in an instance of repeatedly going through these reactions and taking NAD plus to NADH is that NADH can sort of get stuck in a higher ratio than NAD plus. And because NAD plus is so important for fueling the Krebs cycle and glycolysis, this imbalance can upset energy metabolism. And so at high intakes of alcohol, this ratio can be disrupted. If acetaldehyde also builds up as a result of that, then adducts can form where we add other components to the acetaldehyde and then they hang around longer, and that can lead to reactive oxygen species or free radical formation. So switching gears and looking more at NADPH, this cofactor helps with production of new molecules. So here NADPH in fatty acid synthesis is, we're basically taking a longer fatty acid and altering its chain length structure and moving around double bonds. And so we're priming it basically to go into the Krebs cycle. So here we have a 16-0 fatty acid going to a CoA form. So we're just adding CoA, and that allows it to then enter mitochondria for further breakdown because it looks like acetyl-CoA. And we see a couple different steps that utilize NADPH. Now let's look at niacin in reference to interactions with other nutrients. So again, these are biosynthetic pathways, and here we're talking about recycling of other nutrients into their most active form. So one of the best examples of this is through antioxidant recycling. So glutathione, vitamin C, and vitamin E are all tied to each other in sort of a hot potato game of passing electrons from one to the other. And if you go all the way to the beginning of this pathway, you see that NADPH is the first electron donor. So it gives electrons to glutathione, and then glutathione gives electrons to vitamin C, and vitamin C will give electrons to vitamin E. And this is how they're regenerated to their active forms, which are tocopherol, dehydroascorbic acid, and glutathione. Now, recommended daily allowances for this nutrient change with age. So as we get older, we do need more, which makes sense because our bodies get bigger. And we have slight differences between males and females once we reach the teen years. So women need 14 milligrams while men need 16, and then for adults 19 and up, we have that consistent difference. The only time that women would need more than men is when they're pregnant or lactating, and then the demands go up 1 to 2 milligrams more than men. And of course, don't forget that niacin can be produced from 60 milligrams of tryptophan in order to make 1 milligram of niacin. And so this is a way that we do get niacin into our bodies, but is often not accounted for on nutrition facts labels or places that we compute nutritional intake from food directly. And of course, needs can vary from person to person. So some factors that could change someone's needs where they may need more niacin is if someone has a malabsorptive disorder, autoimmune conditions like Crohn's disease or irritable bowel. HIV infection can also increase the need for niacin, and chemotherapy treatment can also alter how much niacin that someone needs. To assess our status, we typically look at urinary excretion of N-methyl nicotinamide. We consider any urinary level greater than 1 milligram per day as desirable, so that implies that you need to collect all the urine from a day to sort of calculate this. And then anything under 1 milligram per day, is particularly once you hit 0.8 milligrams, that's considered poor status, and supplementation or dietary intervention would be needed. We can also look at blood levels of NAD and the NAD to NADP ratio. A normal ratio is 1, so we want to see roughly equal levels of those two metabolites in red blood cells. Typically, if you're looking for circulating niacin, that's just in the fluid part of blood, the plasma, those concentrations remain pretty low under healthy conditions, so it's not really a good indicator. So urine and then red blood cells are the two best ways to look at status. Now, deficiency of niacin is called pellagra, which is an Italian word for raw skin. The signs and symptoms fall into 4 categories that start with D, so dermatitis, diarrhea, dementia, and in very severe cases, death. The dermatitis often manifests as a symmetrical rash, starting on the tongue and the mouth. And so you can see some pictures where it's more advanced and it's moved away from the face and down onto the extremities and the neck and chest. Diarrhea is pretty self-explanatory. It's not always diarrhea. It could be vomiting in some people. And then the dementia can manifest in a variety of ways, characterized as depression, apathy. Some people report headaches and memory loss, and in more severe cases, there could be paralysis. Causes of deficiency are typically poor dietary intake or poor tryptophan sources. So corn is not a good tryptophan source and has obviously all of those binding agents that can reduce the bioavailability of niacin without hydrochloric acid or lime treatment. Certain medications interact with niacin absorption and metabolism. So these would be isoniazid and mercaptopurine. Malabsorptive disorders, so these would be our chronic diarrheal diseases, inflammatory bowel conditions, certain intestinal cancers, and then Hartnup disease. And clinical signs often develop after 50 to 60 days. So keep in mind, we do have daily recommendations for nutrients, but just if you have an off day and you don't get enough niacin on one day, you're not going to have deficiency symptoms right away. You have to repeatedly have low intakes, and it takes about then 50 to 60 days for that to kind of start to have symptoms. Treatment is relatively easy. You give 100 milligrams of nicotinamide 3 times a day for 1 month and then reassess, but typically that will bring the status back up. And there are risks of toxicity, so there is an upper limit of 35 milligrams. This includes nicotinic acid, nicotinamide, and fortified food forms of niacin. There are extended release nicotinic acid supplements if you do need high doses that have fewer side effects, which we will look at now. So if someone exceeds the upper limit, the major signs and symptoms are flushing due to vasodilation, and this particularly happens with nicotinic supplements or nicotinic acid supplements. This is why most supplements have sort of moved to nicotinamide because it's less likely to cause flushing. The flushing or vasodilation is because the influx of niacin causes a really big prostaglandin release, and the severity of that could be lowered with low dose aspirin. And there are, some weightlifting supplements that have really high levels of niacin, and many people who take them do report these sorts of flushing symptoms. And if someone is repeatedly exceeding the upper limit for long periods of time, this could actually injure the liver by ending up blocking the bile duct, from being able to enter the small intestine. People report heartburn, nausea, and vomiting, gout, and high blood glucose levels. And I think this is also a phenomenon that we have seen more of with the increased interest in consuming energy drinks, because many energy drinks contain high doses of niacin. Many of them have moved away from nicotinic acid style niacin, so hopefully this is not a problem. And obviously this would occur once you're kind of drinking multiple cans of these drinks on a daily basis. Now, although we were just talking about toxicity, symptoms, and risks, there are some potential benefits of taking high doses. And so these would exceed the upper limit. Nicotinic acid particularly has been shown to be able to treat hypercholesterolemia. So this is a genetically linked condition where someone has really high levels of LDL cholesterol in their blood, and treating with 6 grams of nicotinic acid a day can actually help to lower that. This happens because the nicotinic acid helps to inhibit the breakdown of adipose tissue, and then therefore decreases hepatic secretion of very low density lipoprotein, the precursor to LDL, so then you get that drop in LDL production. It also inhibits diacylglycerol acyltransferase in the liver, so this is how we break down triglycerides. This is how we produce triglycerides, so this is going to lower our ability to produce more triglycerides. And there are some studies that have shown that high-dose nicotinic acid can help you pass a drug test, and that they may detoxify the body. The scientific evidence on some of those is slim, but there are case reports and other things, and so fully understanding how and why that link was made may not be fully elucidated yet, but those are some other things that have been reported. And then, of course, we have to consider nicotinamide as well, since it acts a little differently than nicotinic acid. There's been topical use of nicotinamide for acne, and then also skin cancers will respond to some forms of treatment with nicotinamide. Specifically, 500 milligrams orally each day can give a reduced rate of new non-melanoma skin cancers in persons with 2 or more non-melanoma skin cancers in the last 5 years. So definitely some kind of cool emerging evidence for high dose use of niacin, but should always be done under the oversight of a medical professional and dietitian combined. But I always think it's interesting to share these things with all of you. So that concludes niacin. So we'll reconvene to look at riboflavin in our next lecture, and I hope you have a great rest of the week.